Good day, everyone. We are Group 9, and our study is entitled Interaction Effects of Temperature and Salinity on the 2R absorption capacity of hydrogel polymers. Our group members are me, Angelica R.J. Maranya, Dale Talavera, Jose Luis Fahad Nilag, and David Zion E. Mangao. In this presentation, we will discuss the background of our study, the methodology we used, our statistical findings, and the recommendations based on our results. Next slide, please. Water scarcity has become one of the biggest challenges in agriculture today. As fresh water becomes less available, many farmers rely on alternative water sources, which often contains dissolved salts. Hydrogels are commonly used in agriculture because they can absorb and retain large amounts of water, helping improve soil moisture and reduce irrigation frequency. However, environmental conditions such as temperature and salinity may affect how much water hydrogels can absorb. If their absorption decreases, their effectiveness in agriculture may also be reduced. Because of this, we investigated how temperature and salinity influence hydrogel water absorption. I now pass the floor for Jose to continue the research gap.
Previous studies have already investigated the effects of temperature and the effects of salinity separately. However, very few studies have examined the interaction between these two factors, especially during the first 2 hours of hydrogel hydration, which is an important period immediately after irrigation. To address this gap, our study focused on determining both the individual and combined effects of temperature and salinity on our hydrogel absorption. Our study used a 2 by 2 factorial experimental design. We investigated two independent variables. The first factor was temperature, with levels of 10 degrees Celsius and 60 degrees Celsius. The second factor was salinity, with concentrations of 0% and 5% salt solution. This resulted in 4 treatment groups with 4 replicates each, giving a total of 16 experimental trials. To analyze our data, we used MATLAB and performed a two-way analysis of variance, or two-way ANOVA. This statistical test allowed us to determine whether temperature, salinity, and the interaction between the two variables had significant effects on hydrogel absorption. We used a significance level of 0.05 for all statistical tests.
And for our expected results, before conducting the experiment, we expected that hydrogel immersed in cold pure water or 10 degrees Celsius with 0% salt would exhibit the greatest water absorption. This expectation was based on previous studies suggesting that saline solutions reduce hydrogel swelling by decreasing the osmotic pressure gradient, while lower temperatures may allow hydrogels to achieve greater equilibrium swelling over time. Therefore, we predicted that increasing salinity would decrease absorption with the 60 degrees Celsius and 5% salt treatment, producing the lowest absorption. And for our experimental setup, to conduct the experiment, we prepared 4 treatment conditions representing the combinations of temperature and salinity. Each hydrogel sample initially weighed 1 gram. The samples were immersed in their assigned solutions for 120 minutes. After soaking, excess water was removed, and the final mass of each hydrogel sample was measured using a digital scale. The collected data were then used for statistical analysis. And for our results, our findings showed a different pattern for our initial expectation. The hot pure water treatment, or 60 degrees Celsius with 0% salt, produced the highest final bead mass of approximately 43 grams. The cold pure water treatment followed with about 38 grams. Both salt water treatments produce much lower values ranging from only 7 to 8 grams. Overall, adding 5% salt reduced hydrogel swelling by approximately 81%, demonstrating that salinity had a much greater effect than temperature.
To determine whether these observed differences were statistically significant, we analyzed the data using the two-way ANOVA. The results showed that temperature had a statistically significant effect. Salinity also had a statistically significant effect, and the interaction between temperature and salinity was likewise significant. This means that the influence of temperature depend on whether the hydrogel was placed in fresh water or salt water. Therefore, both environmental factors must be considered together rather than independently. Our results can be explained by the movement of water molecules through the hydrogel polymer. High temperatures increases the kinetic energy of water molecules, allowing them to diffuse into the hydrogel more rapidly during the limited 2-hour soaking period. However, salt creates a hypertonic environment that reduces the osmotic pressure driving water into the polymer network. As a result, saline conditions greatly restricted hydrogel swelling regardless of temperature. This explains why salinity became the dominant factor affecting hydrogel performance.
In conclusion, our study demonstrates that both temperature and salinity significantly influence hydrogel water absorption. Among these factors, salinity was the strongest limiting factor, while higher temperature accelerated water absorption only under freshwater conditions. A significant interaction between temperature and salinity was also confirmed through statistical analysis. For future studies, we recommend extending the immersion period, evaluating additional temperature and salinity levels, using closed containers to reduce evaporation, and testing agricultural-grade superabsorbent polymers to better represent actual farming conditions. These are the reference used in conducting our research paper. Next slide, please. And more references that was used. That concludes our presentation. Thank you very much for your time and attention. We hope our study contributes to a better understanding of hydrogel behaviors under different environmental conditions and provides useful information for future agriculture research and water conservation practices.